kind of fun stuff she has in store for you. So chapter 6. This quote is by a guy named Edward Abbey. You guys probably don't know who that was, but back in the 1960s and into the 1970s in the United States was a time of greatly increased awareness of the impact of human activities on the environment, and what we now refer to as the environmental revolution. And there were a lot of well-known activists, environmental activists and authors at that time, and he was one of them, Edward Abbey. He wrote a book, several books, but one well-known book he wrote was called The Monkey Wrench Gang, and it was about about environmental, well, I guess we would describe as environmental terrorists. These were environmental activists who would like sabotage logging equipment, like because they objected to cutting down forests, so they would go to where the loggers were cutting down the forest, and they would sabotage the bulldozers and the logging equipment. What's that? Yeah, put spikes in trees so that so that the chainsaws would hit that and would mess up the chainsaws. So that was called monkey wrenching. But this guy was kind of a character. He was a kind of a hippie type, bearded, kind of scruffy looking guy. And I read that he was buried in his pickup truck. He wanted to have his pickup truck as his coffin. But this quote, you guys know what that means. He's referring to or comparing. He's comparing cancer, which is there are many different forms of cancer, but cancer is a type of disease that involves cells dividing out of control. So normally cells of our bodies are only divide at certain times. Some cells divide just about constantly, like your skin cells are always dividing to make more because your skin cells are always rubbing off or falling off. But other kinds of cells, like the cells of your skeletal muscles, they never divide. Once the skeletal muscle cells that you have when you're born, that's all you get for the rest of your life. If you lift weights and your muscles get bigger, it's not because you're gaining more muscle cells, it's just the muscle cells that you have are getting thicker and stronger. So he's comparing this out of control growth of cancer cells to through the growth of cities and development of natural areas into cities and parking lots and shopping centers, which some people consider that to be progress, but other people consider it to be just growth for the sake of growth. Should we be building, expanding cities just because we can, building more buildings just because we can do that, or we want to do that? Okay, so let's look at this video. Might as well.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Okay, so that's what we're going to be learning about in this chapter. Type of cell division called mitosis.
Introducing a completely reimagined disc and an all-new Golf Galaxy Performance Center. For a sports destination unlike any other. With an expanded assortment of the latest styles and the best gear that takes your game to the next level and beyond. Plus, interactive experiences and expert services. Welcome to your new
Okay. A couple of terms here, genome and gene. The term genome just refers to all the genetic information and all the chromosomes in an individual. And so we use the term genome for one individual, but the term gene pool is for a whole population. It would be all the genetic information in all the individuals of a population. So a chromosome is essentially a long DNA molecule. These are very long. It can be, the chromosome can be a couple inches long. It's all coiled up inside of a cell. And it's a chromosome is made up of genes. So each section of a chromosome is called a gene. And a gene, so the gene has the information for a cell to do something. And usually what what the information is for making a protein molecule. So it's how to put the amino acids together in what order to make a particular type of protein molecule. And then the proteins do all the work in the cells. Okay, in eukaryotic cells like humans, there are some number of chromosomes that so that they're linear, which just means they're long and straight like this. Whereas in prokaryotic cells like bacteria, there's usually just one long chromosome that's in a loop or a ring of DNA, and there are usually some. So within a bacterium, there's usually could be one big loop-shaped chromosome, and there'd be some small ones in there too, called plasmids, which are usually just one gene on a little loop of DNA. Okay, what's shown here is a chromosome. This is a cell. Here's a nucleus. Here's a bunch of chromosomes. In humans, human cells, there would be 23 pairs of chromosomes in that nucleus. So each chromosome is a long DNA molecule, and along this DNA molecule there are protein molecules called histones spaced out along the DNA, and the DNA along with the histone proteins on them is called chromatin. Chromatin. So we would say that chromatin is the material that the chromosomes are made up of. DNA with histone proteins attached. And the function of these histone proteins is to coil up the DNA molecule like a spring, you see here. And then this coiled DNA, it coils up again, and so the result is instead of this DNA molecule being very long and strung out, it's short and thick enough that you can see them under a microscope. When it's just uncoiled like this, it's so fine that you can't really see a DNA molecule with a light microscope, a compound microscope, when it's uncoiled. But when it's coiled, short and thick like this, they're visible. But they only do this. They only coil up like this when the cell is going to divide. Because when the cell divides, the chromosomes separate into the daughter cells, and that would be hard to do if these DNA molecules were all strung out like this. But when they're short and thick, it's easy for them to move around during cell division. This looks like a letter X here, and that's because another thing that happens when a cell is getting ready to divide is it makes a copy of each of its chromosomes. So here's a cell that's dividing. So in cell division, we would call this the parent cell, and then these the daughter cells. So if this is a human cell, it has 23 pairs of chromosomes. When the cell divides, half of those chromosomes go into one daughter cell, the other half go into the other daughter cell. In order for these daughter cells to have the right number of chromosomes, which is 23 pairs, these chromosomes are copied before the division so that there's enough chromosomes for both daughter cells. So that's why it looks like this. Each of these 23 pairs of chromosomes in here has been copied, and the two, so the two, we call them sister chromatids like this. They're identical to each other. They're still attached. They'll only separate when the cell is dividing. Okay. The term somatic cell just refers to the cell, to cells that are not the sex cells, they're not sperm or egg. So the sperm and egg are called sex cells or reproductive cells, but all the other cells in your body are called somatic cells. And so somatic cells have 23 pairs of chromosomes. A sperm and egg have only one set of chromosomes, or that is, one set of 23 chromosomes instead of 23 pairs. And that's because later, when there's fertilization, a sperm with one set of chromosomes and an egg with one set of chromosomes comes together, and now that fertilized egg will have the normal two sets, or 23 pairs of chromosomes. But in this chapter, we're talking about somatic cells dividing. There are some somatic cells of your body that don't have chromosomes. An example is red blood cells. Red blood cells, as soon as they're formed, the nucleus disappears, and so they have no chromosomes in them. So red blood cells can't do very much. They don't have any information. They don't have a nucleus with information to do anything. All they do is carry oxygen, and they live a few months and then they die. They can't even reproduce. They can't even divide to make more. This term karyotype just means all the chromosomes in a cell that are spread out and the chromosomes paired up like this. So this is a karyotype. This is what somebody in a lab has done. They've broken a cell open, taken the 23 pairs of chromosomes and put them together. The two number one chromosomes together, the two number two, two number three, and so on. And this image of all these chromosomes from a cell is called a karyotype. This is done a lot in cases of a woman is pregnant, she has an amniocentesis or other processes of getting cells of the fetus and to do a genetic test on them. So amniocentesis is putting a needle through the body wall into the amnion, the sac that surrounds the fetus, and withdrawing some of that amniotic fluid, and that surrounds the developing fetus. And that fluid will have some fetus cells in it. And so they do a karyotype like this with the fetus cells. And you look at them, and you might want to just make sure there's actually 23 pairs of chromosomes here. If there's a chromosome missing, that's not good. Or some cases they might notice that there's 3 number 21 chromosomes. That's not right. Anybody know what that would mean? If a fetus has 3 number 21 chromosomes. Yeah, you're right, Down syndrome. That's called trisomy, the condition of having 3 of a chromosome is called trisomy. So trisomy 21 is the cause of most cases of Down syndrome. 95% of Down syndrome cases are causes trisomy 21. But these chromosomes are numbered by length. The number 1 chromosomes are the longest, and then these are number 2 is a little shorter, and so on. So these are all this. So these are this chromosome that says X, and the 23rd pair of chromosomes are the sex chromosomes, the X and the Y. So a human, he has either 2 X chromosomes or an X and a Y. So the Y chromosome is much smaller than the X. These are about the same length, so this would be XX. So that would be what, female or male? XX. 2 X's. No, it's female. If it was XY, that would be a male, right? So the X and Y are called the sex chromosomes. All the others are called autosomes. That's the first 22. And so each one of these chromosomes has a bunch of genes along it. And the chromosomes of a pair are called homologous chromosomes. So these two, we would say that these 2 number 7 chromosomes are homologous. That's a homologous pair. Homologous means that they're comparable to each other, kind of the same, but not exactly. For example, on these 2 number 1 chromosomes, if there's a gene for eye color right here on this number 1 chromosome, then there will be another gene for eye color in exactly the same place on the other number 1 chromosome. And that location on a chromosome is called the locus. The genes at a particular locus on 2 homologous chromosomes are the same kind of gene, eye color gene, for example. But the one on this number 1 chromosome could be for blue eyes, and the other one could be for brown eyes. But they're both eye color genes. They could be different, or they could be the same. We'll talk more about that when we do the genetics chapter. But so the term homologous pairs. So 22 pairs of autosomes and 1 pair of sex chromosomes, the X and Y. Some people have an extra X or Y chromosome, or have only 1 X chromosome, but those are abnormalities. This term allele just means it means the same as genes. Alleles are just genes, but there can be different alleles for a type of gene. So in the case of eye color genes, we know there's a blue eye allele and a brown eye allele for that type of gene. There's another karyotype. This one is a male, but there's X and Y instead of 2X. You'll see the Y chromosome is much shorter. So we would say these are not homologous. That's an exception. This pair is not homologous. They're not the same. They don't have the same kind of genes on them. And why? What's on that Y chromosome? Or what's on the X and Y chromosomes? There are a lot of genes on the X chromosome that have nothing to do with the difference between male and female, but on the X chromosome is the information for developing the reproductive organs, and so everyone has an X chromosome. But some people have 2 of them. Some people have only 1 and a Y chromosome, depending on if you're female or male. So if you're female, if you're either male or female, as your reproductive organs start to develop, they'll be using the information in the X chromosome to develop. And so for the first several weeks of development of an embryo, whether you're male and female, your reproductive organs will develop exactly the same. Can't tell them apart, whether you're male from female. But if you're male and you also have a Y chromosome, then at some point in your development as an embryo and fetus, the information on the Y chromosome will redirect the development of the reproductive organs, so that instead of just continuing to develop as the female reproductive organs, the uterus and ovaries and so on, they will be modified to develop into the testes and other male reproductive organs. I've used to put extra credit questions at the ends of some of my exams, and one time I guess I did this a few times. I put a question: name a person who has no Y chromosome, and most of the students in the class would leave it blank. They didn't know how to answer that. When it's the simplest question they could ask. It's any female. Just name any female would not have a Y chromosome. Well, one answer I got several times, people put Michael Jackson. But he has no Y chromosome. I guess just because he's kind of a weird person, or he was a weird guy. That's who came to mind. Okay, cell cycle. The term cell cycle just means the life of a cell, and all the events that happen in the life of a cell, we call the life cycle. So we would consider when this division occurs to form these two daughter cells, that would be the beginning of the life of these cells. Then sometime in the future, if when this cell would divide, now that would be the beginning of the life of these cells, but we would consider that to be the end of the life of this cell. So the life cycle is from the time of this division, which forms this cell, until the time of this division when this cell divides. So how long that is, how long of a time that is, depends on what type of cell it is. Some cells, I was saying earlier, divide very frequently. Skin cells, the skin cells divide constantly to make more. Cells of the epidermis, the outer layers of the skin. Red blood cells themselves don't actually divide, but we have cells in our bone marrow called stem cells whose job is just to divide to make red blood cells. But those are constantly dividing because we have many billions of red blood cells in the body, but they're dying all the time. They only live 4 months and then they die, so we have to always keep making more. But others, like I said, skeletal muscle cells, once they're formed, they don't divide to make more. Most of the nerve cells, neurons, the nerve cells of our central nervous system, the brain and spinal cord, they don't divide. So that's why if you have a spinal cord injury or a traumatic brain injury and you lose a lot of those nerve cells, you don't get them back. Because your remaining cells don't divide to replace them. Some cells will divide only as needed. So if you have an injury or an infection, something that kills or destroys some of the cells of some tissues of your body, then part of the healing process is for the cells to divide to replace them, the cells that have lost. And this is all programmed into our cells in the DNA. When your cells should and should not divide are in our chromosomes. We have genes just for that, just to tell our cells when they should divide and when they should not divide. And it's when those genes that regulate cell division, when those genes get mutated or changed, that's when cancer happens. Now your cells don't know when they should. They'll just they'll think they should divide, and then divide and divide, and they'll just don't know when to stop. And that's what causes cancerous tumors, and these cancer cells never become functional, normal tissue cells. They're just cancer cells. All they know how to do is just keep dividing and disrupt the normal function of the tissues or organ in which this is occurring. Okay, the cell cycle can be divided into two parts, interphase and M phase. M phase is when the cell is dividing. So M would stand for mitosis, or mitotic cell division. So interphase is just the rest of the time, when the cell is just living its life and existing as whatever type of cell it is. And interphase would also include the time when a cell divides, if a cell decides that it's going to divide, and then it gets ready to divide. So here's a diagram showing the cell cycle and the different stages of the cell cycle. So here's the M phase. So this is when the cell's dividing, and this is interphase the rest of the time. So the term mitosis, the real definition of mitosis, we use that word all the time to mean cell division, but the really correct definition of mitosis is division of the nucleus of the cell. It's in mitosis where the chromosomes separate into the daughter cells. And the division of the rest of the cell is called cytokinesis. A better term for this kind of cell division would be mitotic cell division, which includes mitosis, which is division of the nucleus, and cytokinesis, which is division of the rest of the cell. So after a cell divides, would be the beginning of the life of a new cell. And after that is a phase, the first part of interphase, which is called G1. G stands for growth. So this is a period after a new cell is formed. So if a cell divides, then these two daughter cells are each about half the size of the parent cell. And so these are going to have to grow to get to be the normal size for that type of cell. So that happens in the G1 phase. There will be more organelles forming, so that these daughter cells will have enough mitochondria and lysosomes and chloroplasts if it's a plant cell, and so on. That'll all happen in the G1 phase. And it also has this going off to the sides, this stage called G0. And this just would be if the cell just now, after the G1 phase, if it just goes on and exists for years and years and years, and just living as a cell in your body, that would be what we call G0. It has not just divided, and it's not getting ready to divide. It's just living its life as a cell. And for some cells, like your muscle cells, this will be the rest of your life because they never divide. But in some other cells, this could be very short period of time. And in other cells, it might be just until something stimulates that cell to divide. But now if the cell decides it's going to divide, then will be the S phase. S stands for synthesis. Synthesis means making something. And what's being made is a copy of every chromosome. So here's where the chromosomes are duplicated or replicated because the cell's getting ready to divide. And there has to be enough chromosomes for the two daughter cells when it divides. So all that DNA is being copied. The DNA of every chromosome is being copied. And then right before the cell divides, it goes into another growth phase called G2. So before the cell divides, it'll grow a little bit, get a little bit bigger. So and maybe it'll make some more organelles so that each of the daughter cells will have enough organelles to get started in their lives. Okay, so this is what I was just saying. The phases. Interphase includes G1, S, G2, and G0. So here's again what a duplicated chromosome looks like. There was one chromosome, and then when the cell decided that it's going to divide, it would be copied. So now there are two identical chromosomes. We call them sister chromatids until they separate from each other during cell division, and then they'll be called chromosomes. And these are short and thick. They appear short and thick because they've coiled up, getting ready for the cell division. Okay, so now we got to look at what happens in cell division, or the mitotic phase. Exactly how does a cell divide? So again, that involves mitosis, which is division of the nucleus, and cytokinesis, which is division of the rest of the cell. So first of all, this kind of mitotic cell division is described as duplication division because these daughter cells should be exact duplicates of the parent cell. They should have, that is, they should be genetically identical. So they should have the same 23, in the case of humans, you should have the same 23 pairs of chromosomes, the same genetic information as the parent cell. When we look at this other kind of cell division called meiotic cell division, or meiosis, in the next chapter, we'll see that it's a little bit different. That kind of cell division is only for producing sperm and egg. It only occurs in the testes to make sperm or in the ovaries to make eggs. And that's not duplication division because the sperm and egg are not identical to the parent cells that divide to produce them. But we're just talking about mitotic in this chapter. So if you're looking at cells under a microscope, you might see some that look like this and some that look different like this. On this cell, you can't, so here's the cell, here's the nucleus, and you can see a bunch of stuff in there, but you can't really see individual chromosomes. You just see all this dark stuff. So that would just be all these chromosomes are just strung out in here. And and you can't really see individual chromosomes. But here, this one cell looks a lot different. This one looks a lot different. These are cells that are in the middle of dividing. And we'll see in a minute exactly what they're doing. So this mitosis involves organelles called centrosomes. It says they're the microtubule organizing centers of the cell that produces the spindle apparatus. This spindle apparatus just refers to microtubules, which are just protein, long protein fibers that attach to the chromosomes and move the chromosomes around. So the chromosomes have to be separated into the daughter cells, and that's what the spindle apparatus does. So there's normally, when the cell's not dividing, there's one centrosome, but when during the S phase, when all the chromosomes are copied, the one centrosome will also be copied. So now there's two centrosomes at the time of cell division. There has to be two of them. They'll be at either end of the cell to pull the chromosomes toward the opposite ends. So here's a summary. We'll look in a minute in more detail, but here's a summary of mitotic cell division. So here's a cell that's going to divide. The cell and the nucleus here with chromosomes, and there's only two pairs of chromosomes in here. We see two short ones and two long ones. If this was a human cell, there will be 23 pairs. But to make it simpler, there's only two pairs in this diagram. Different species of animals and plants have different numbers of chromosomes. In humans, it happens to be 23 pairs. In a horse, it might be 27. In a dog, it might be 21. So in this cell, there's only two pairs. That's what this means here. This 2n equals 4. N equals the number of different kinds of chromosomes. So there's two. In this case, there's two sets of 2 chromosomes. There's a total of 4. If this was a human cell, n would be 23. So 2n will equal 46 total chromosomes. So cell's getting ready to divide, so it's going to copy all of its chromosomes during the S phase of interphase. So now all the chromosomes have been copied, two sister chromatids still stuck together here. This place where they're connected to each other is called the centromere, where the two sister chromatids are still stuck together. Then the division occurs, mitosis division occurs. Now there's two daughter cells. Each daughter cell got one sister chromatid from each duplicated chromosome. And now the two daughter cells are exactly like the original parent cell. There's two sets of chromosomes, two pairs of two chromosomes. We'll see next week when we study genetics that these chromosomes are colored red and blue here. You got 23 pairs of chromosomes. For each pair of your chromosomes, one of them came from your mother and one came from your father. So we could say that the red ones are from this person's mother, and the blue ones are from the person's father. But when these sister chromatids separate, never mind. Sorry. we don't need to talk about that yet. It's confusing. Sorry. I'll wait to talk about that. Okay, some practice questions here. Now, the phases of mitosis, or the different steps of the mitotic cell division, are listed here. And this lists 5 of them. Most biology books list only 4. This book that we're using now, I don't know why. Some books have an extra one they call prometaphase. In that video we looked at before, it was mentioned that prophase includes prometaphase. Some books never mention prometaphase at all, and some of them have it as a separate step. But, the way biology students would typically remember the steps of mitosis in order with PMAT. Prophase, metaphase, anaphase, telophase. But now there's in our book there's an extra one, so it would be PMAT. If you include interphase in the life cycle, then it would be IPMAT. So we use this textbook we use in this class. It's not the best. We wouldn't have chosen this textbook except for the fact that it's free online. It's an OpenStax textbook. The book that we used before we switched to OpenStax, you'd have to pay $200 for that book. So we did it to save students money. After this semester, we just found out last week that the RCC bookstore is going to just about shut down. The bookstore is not going to sell books anymore. Just going to sell, I guess, T-shirts and pens and pencils and notebooks and stuff. So we're not allowed. Instructors are not allowed to have students buy anything. We're not allowed to have you buy a textbook or anything that costs money. So for the science department, we've already switched to OpenStax, so that's not going to affect us anyway. We just tell you just access the book online. It's free, but all the other departments haven't done that. They're going to have to come up with online books or something. What's that? But I don't know. I've had some instructors already complaining that there is no free textbooks out there, and so we don't know. They don't know what they're going to do. They're going to have to make photocopies of stuff or come up with stuff themselves and put it on Moodle for students to read. Okay, so here's the steps of the stages of mitosis and what happens in each one. So up here we've got diagrams or drawings. Down here these are micrographs or photographs through a microscope of cells that are dividing. So we need to know what's going on in this, and learning the steps of mitosis is something that it's one of those things that biology students don't like to do. But it's not that hard if you understand what's going on. It all makes sense. So here's prophase. So well, it says chromosomes condense and become visible. So the chromosomes have coiled up. That's what condense means. They've coiled up. So now they're short and thin. They're visible under a microscope. The spindle fibers emerge from the centrosomes. So here's two centrosomes, and you see these lines. They're blue lines coming out. So those are the spindle fibers coming out. Those are going to attach to the chromosomes. So the nuclear envelope breaks down, or so the nuclear envelope disappears. It has to do that because the chromosomes are going to separate into the daughter cells, and they could not do that if they were surrounded by the nuclear envelope. And these two centrosomes, they're moving toward opposite ends of the cell, of the poles, the opposite ends of the cell. So in the next step, prometaphase, so the chromosomes continue to condense, so the chromosomes are getting shorter and thicker. These kinetochores appear at the centrosomes. Remember, the centrosomes are where the two sister chromatids are attached together, and the kinetochores are just the attachments for the spindle fibers at the centrosomes. The metaphase, so the chromosomes are lined up at the metaphase plate. The metaphase plate is just an imaginary plate down the middle of the cell. That's also sometimes called the equator. It's just the center of the cell, just like the equator around the midpoint of the Earth between the north and south poles. Not the midpoint, but the circumference. So just remember meta-middle. The chromosomes are all lined up in the middle during metaphase. And each sister chromatid is attached to a spindle fiber. So all these spindle fibers from these from the centrosomes have extended out and attached to these kinetochores at the centromeres of those duplicated chromosomes. So under a microscope, if you see a cell and all the chromosomes are lined up in the middle, or it might just look like a dark line down the middle of this cell, that's obvious. That cell is dividing and it's in metaphase. Now in anaphase, the sister chromatids have separated from each other, so they're being pulled apart by these spindle fibers. And so you can see a little bit of daylight in between two lines of chromosomes now. So if you see, this is kind of weird looking. It's like they're glowing, but you see two lines of chromosomes with some space in between them. That would be anaphase. And telophase, the last step. Now these chromosomes, two sets of chromosomes have moved all the way to the opposite ends of the cell. So now there's two sets of chromosomes. If this was humans, there would be two sets of 23 pairs of chromosomes at opposite ends of this cell. So the chromosomes begin to decondense. So these chromosomes will uncoil now. A new nuclear envelope will form around each set of chromosomes, so a new nucleus. The mitotic spindle, these spindle fibers will break down. They're not needed anymore because their job was to move the chromosomes, and they've done that. And usually at the same time as telophase, cytokinesis occurs. So cytokinesis is the rest of the cell dividing in half. And so we see the cell pinching in half there. This groove that forms down the middle of the cell, pinching it in half, is called the cleavage furrow. In the case of plant cells, the plant cells have a cell wall around them, so they can't pinch like that. They can't they're stiff, so because of the cell wall, so they can't pinch in half. Instead, it'll form a new cell, a new wall of plasma membrane and cell walls down the middle here. That's called the cell plate. So that's how. So that cell plate divides the two daughter cells in half in the case of a dividing plant cell. So now these cells, two daughter cells will be separated, and each one will should be identical to the original parent cell. Talking about cell divisions, one of the purposes of cell division is growth, right? There's a lot of cell division going on as we're growing, before we're born, and then throughout childhood we're growing. A lot of cell division going on, and that's how do those cells know that they should be dividing for growth? It's because of hormones. There's pituitary gland in the brain produces growth hormones that tell your tissues of your body that the cells should be dividing for growth. Question, what's the fastest growing tissue in the animal kingdom? Skin? No. There's only one group of animals, or one family of animals, that has this kind of feature, this tissue.
Is it an animal, or you said mammals?
It's an animal. It is a mammal, though. It is in a mammal, or a group of mammals. And there's one species in the Carolinas that has this kind of feature. It is, you're right, antlers. Deer antlers. Antlers are only in animals of the deer family. So the white-tailed deer that we have around here, and the mule deer out west, and the elk and caribou and moose, those are all. So that antlers can grow as much as an inch a day in length while they're growing. So most deer species, they drop their antlers every year and grow a new set. And so it takes a lot of energy and nutrients for a deer to do that every year. It's white, and it's only the males in most species. In the caribou, it's both males and females have antlers. But what do the male deer do with those antlers? Why do they need them? Defense. Sometimes for defense, yeah. What else? For what other reason is it important for a male? Mating, yeah. That's sexual selection. So the female deer prefer the males with large antlers. That's attractive to them. There was a species of deer that's extinct. It went extinct about 8,000 years ago or something. It's called the Irish elk. So it was in Europe, and this it's kind of like a moose, a large moose, but its antlers were so huge that its rack of antlers could be 10 feet across. The antlers on that animal weighed more than the whole skeleton of the animal, and some scientists think that maybe that's why they went extinct. The antlers got bigger and bigger because the females prefer large antlers. And so the males with the larger antlers would be the ones who would get to mate with the females the most. And so the antlers, just each generation would get a little bit bigger for that reason, until they got so big that the antlers would just made it harder for them to survive, because it'd be hard to run from a predator. Back then there were like saber-toothed tigers, big predators that could take down a big deer like that. But it'd be hard to run with a rack of antlers that size on your head, especially running through a forest, trying to run between the trees. So what are antlers made out of? What is that tissue? Huh? Not antlers. No, that's what horns. Horns are keratin, like a rhinoceros horn or the horns on giraffes. That's keratin protein. But antlers that's just solid bone. So when I said that only deer have that tissue, I was kind of lying. All mammals have bone, but that's starts out soft like cartilage and then hardens into bone later. Okay, where were we talking about this? Okay, so why is mitosis, cell division, necessary for growth and repair? Healing and replacing old dying cells. In the case of certain types of cells that just only live a certain period of time and then they just naturally die. Then the term for cell dying is apoptosis. Apoptosis. That's defined as programmed cell death. So cells like red blood cells, their lifespan is just 4 months. So they just normally die after that period of time. So that would be a type of apoptosis. Okay, some practice questions. Okay, I already talked about cytokinesis. So this happens usually at about the same time as telophase. Telophase being the last step in the mitosis, or the division of the nucleus. The rest of the cell will be dividing. In the case of an animal cell, it just pinches in half with a cleavage furrow. In the case of a plant cell, a cell plate forms down the middle. So animal cell just pinch in half, but in a plant cell, you can't do that because it has a stiff cell wall around it. So instead, it forms a cell plate. So that will be a new layer of plasma membrane and cell wall to separate the cells. So here's what under a microscope what a cell would look like that is just divided. It's almost completely divided. So this is an actual micrograph. And this would be a micrograph of the cell plate forming with, between two daughter cells in a dividing plant cell. So the cell plate starts out as just a bunch of vesicles. So these vesicles are made of membrane material, so they just come together and fuse together and form a continuous new plasma membrane there. And then a new cell wall would form. in between the plasma membranes. Okay, mentioned cancer earlier. We're going to talk more about cancer and what your cells try to do to prevent cancer happening. So, I was talking earlier about how do your cells know when they should and should not divide? There are genes in your cells just for that, just with the information. And some of these genes tell your cells when they should divide, and others tell them when they should not divide. So if there is an accidental change, which in any of those genes, and the term for that is mutation. A genetic mutation is just an accidental change in a gene. Something happens that changes the nucleotide sequence, the series of letters, changes that by mistake. And now that gene is not normal, and now that gene doesn't tell the cell when it should divide or when it should not divide. So now maybe the cell thinks it should be dividing now. So it'll just keep dividing. It'll divide. Now the daughter cells will inherit that same mutated gene, and so they'll divide when they're not supposed to, and so on. And that's cancer. And it usually takes more than one of those mutations for a cell to become cancerous. So we accumulate those mutations in cells throughout our lives. Some people are born, you can be born or inherit one of those cancer genes from your parents. And that makes it more likely that you'll get cancer, because now if you just get one or two more in a cell, then that cell will become cancerous. There's such a gene for breast cancer, and that we know is inherited. And so if a female, so if you are female and your mother had breast cancer and your grandmother had breast cancer, then good chance you've inherited that gene for breast cancer. But anyway, all these things that happen in the life cycle or in the cell cycle, all those things that happen, like the S phase, when all the chromosomes are copied, that's an opportunity for a mistake, a mutation to happen, because each one of those 23 pairs of chromosomes has to be copied exactly. All of those letters, and there's billions of them in your genome. All those letters have to be copied exactly in the right order. If one of them is the cell puts a C instead of a G somewhere in one of your chromosomes, that's a mutation. Some mutations are harmless, they don't really make a difference, but in some cases, just one letter, one nucleotide being wrong could be devastating. When during mitosis, when the chromosomes are separating into the daughter cells, what if a mistake happens? Like if the two sister chromatids don't separate like they're supposed to, and they would both go into one cell, and the other cell doesn't get any of that chromosome. That would be another type of mutation. But so after many of these stages in the cell cycle, after your cell copies all of its chromosomes, there's what's called a checkpoint, which means that the cell checks to make sure that it did that correctly, that it copied all the chromosomes correctly. And so it might compare the two sister chromatids nucleotide by nucleotide, letter by letter. Do they match like they should? And if it finds a mistake, it'll try to fix it before proceeding to the next stage in the cell cycle. So here's some of these checkpoints labeled on here. So there's a G2 checkpoint, a G1 checkpoint, an M checkpoint. So these are places where the cell checks what it has just done. Like the M checkpoint, after mitosis, it'll check to make sure that, or during mitosis, it'll check to make sure that both sets of chromosomes have 23 pairs, and that they all separated properly, like they're supposed to. So G1. So is the cell large enough? So this is the one right at the end of G1, the G1 phase. So is the cell large enough? So it have enough nutrients? Any damaged DNA? If it finds anything wrong at that point, the cell will not let itself proceed to the next stage. G2 checkpoint, have all the chromosomes been copied accurately in the S phase right before that? M checkpoint, during metaphase, are the chromosomes aligning properly? And then there's that term apoptosis that I defined already, programmed cell death. So in some cases, if the cell finds damage that it cannot repair, it'll commit suicide. That's a type of apoptosis. So because if the cell cannot fix a mutation, now that might be a mutation that would now make that cell cancerous, a cancer cell. And now if that cell divides, now you've got 2 cancer cells. If they divide, now you got 4 and 8 and 16, and now you've got cancer. But so the cell will sometimes just commit suicide just to save the rest of your body. It'll sacrifice itself. These checkpoints and apoptosis don't necessarily always happen, because if these don't work, then that's how we get cancer. And in the case of mitosis cell division, if one of these mistakes happen when one cell is dividing, that might not be any big deal, because now you've just got one cell, or two daughter cells maybe, that have that mistake in them. But if this happens during meiosis cell division, when you're making sperm and egg, now that mistake or that mutation is in a sperm cell or an egg cell. Now you're passing it on to your kids if that egg is fertilized, or the sperm fertilizes an egg, and now that fertilized egg has that mutation in it, and that mistake that happened during meiosis cell division, and that mitosis could be devastating. It could be it could mean that fetus won't even survive, and there will be a miscarriage, or could be that baby could be born with Down syndrome or some other abnormality. Okay, just another diagram showing the checkpoints and what they do. So this is pretty amazing that this is going on in your cells all the time, and there could be many times in your life when when a cell in your body becomes cancerous, but that cell will just commit suicide and, to prevent development of cancer in your body, and you would never even know it. Okay, more about cancer. This is kind of important. It's defined as uncontrolled cell division. This term carcinogenesis the development of cancer. If you ever see Genesis in the word in biology, it means, it's like in Genesis in the Bible, the origin or the formation of something. So this root, Latin root carcin means cancer. So, like I said, it's caused by mutations in some of the genes that regulate cell division, so that now the division of that cell is unregulated. It just divides out of control when it's not supposed to. So these cells, these cancer cells, they don't become normal functioning cells. A pathologist who is a scientist who studies diseases or diseased tissues can look at cells under a microscope and tell if they're cancerous or precancerous. They're on their way to becoming cancerous. They'll have abnormal nucleus, and they can see that. So if you ever have any kind of tumor or a mole removed or something, any tissue that's removed from a patient in any kind of surgery, that normally that goes to a pathologist to look at it and just make sure that whether it's cancerous or not. Okay, the term carcinogen just means any agent in the environment that causes cancer or makes it more likely that you get cancer. And we know that there are many chemicals that are like that, and certain kinds of radiation, like ultraviolet radiation, and some viruses will deliver cancer genes into cells, into a person's cells. Ultraviolet radiation from the sun, how does that cause? Why is that a carcinogen? It's because, well, it's a higher energy level radiation than light, than visible light. So when that radiation hits your surface of your skin, that can penetrate into your cells, and that energy can hit your DNA in your chromosomes and break the DNA molecules, and that would cause a genetic mutation. The molecule breaks in half, or a chromosome breaks in half, or sometimes a chromosome will break in half, but then it'll come back together, but with a nucleotide missing, or something. That would be a mutation. And because your skin is on the surface of your body, it receives more ultraviolet radiation than any of the deeper tissues in your body. That's why skin cancer is one of the more common kinds of cancer. And also because your skin cells are dividing all the time, and every time a skin cell divides, that's an opportunity for one of these mistakes to happen, a mutation. And so if your cells are dividing while ultraviolet radiation is hitting them, that could make one of those mutations more likely to happen. So, good reason to wear sunscreen and use sunscreen and wear a hat to go outside. Although it's good to have about 15 minutes of direct sun exposure to your skin, at least 3 times a week for production of vitamin C. But too much ultraviolet is bad for your skin. Other types of radiation, X-rays. That's a higher energy than ultraviolet, so you don't want to get too many X-rays. When you have an X-ray, they'll look one part of your body. They'll often put that vest or that thing to cover your much of your body, especially your reproductive organs. You don't want to have genetic mutations happening in the sperm and eggs that you're producing, because you could pass them on to your kids. A lot of chemicals. Um, oh, a lot of products have on them have a label on them or something. This product contains chemicals that are known to cause cancer in laboratory animals or something. Um, Especially in California, they have very strict regulations about carcinogens and labeling products and testing for carcinogens. Um, uh, we have government agencies that are supposed to protect us from harmful chemicals and stuff, like the Environmental Protection Agency, the EPA, or the OSHA, the Occupational Safety and Health Administration, and NIOSH, the National Institute of Occupational Safety and Health or something. These are agencies with with employees who go into factories and stuff and make sure, and they look for unsafe conditions or unhealthy conditions, and so employers are required to protect their employees from stuff like carcinogenic chemicals. And, but really, if you work in a factory or you work in some kind of profession in which you are potentially exposed to harmful chemicals, it's really your own responsibility to take care of yourself, because your bosses are not watching you all the time, and they're not going to. I mean, if they give you earplugs to wear on your job, and you work in a factory where there's very loud machines all the time, if you don't wear those earplugs, that's on you. That's your own fault. I've seen that. I used to work in factories and work with guys. They would not wear earplugs or ear covers in a factory there that was so loud all day long. You couldn't even talk to somebody 2 feet away, and they couldn't hear you. And these guys would walk around in there 8 hours a day with no earplugs, no ear protection. By the time they're 40, they're half deaf. They've lost half their hearing. And it's their own fault. The employer gives them earplugs, but they just don't use them. I remember one. I used to work in the General Electric jet engine plant in Evendale, Ohio, and they had these machines. That it was a machining operation that was making part of the jet engines, and this operation happened submerged in oil on this machine. And the machine operator, after this operation, he'd have to take the part out, and it would be covered with, this part would just be covered with oil from this machining operation. There'd be like a little crane from the ceiling that would, with a chain, and he would hook that onto the part to lift it out of the machine, and it'd have oil dripping off of it. And then he'd move it over and put it into this tank of solvent. This stuff was called trichloroethane, this solvent. And he'd put it in there to rinse the oil off of it, and then it would lift it out of there and put it on a table to drain. And it said a great big symbol, hazmat symbol on this tank of this solvent. It said, danger, causes cancer, do not contact with skin. And this guy, I watched this happen all the time. This guy, at lunchtime, he'd dip a rag into that tank of solvent and clean his arms and hands with it. And he'd do that at the end of the day. This stuff says right there, it causes cancer. That stuff like that will absorb right through your skin, into your tissues. And, so, guy like that, if he gets cancer, it's his own fault. Got to look after yourself. Okay, a gene that has become cancerous is called an oncogene. So this would be one of those genes that regulates cell division, and now it has had a mutation in it. So now it is no longer regulating cell division. So the cell division is happening out of control. So one of those genes that regulates cell division is called a proto-oncogene. So that's a gene that can become an oncogene if it's mutated. Yeah. So there are some genes that tell your cells when they should divide, and there are other cells that tell your cells when they should not divide. And those genes are called tumor suppressor genes. The genes that normally tell your cell when it should not be dividing. And if one of those genes gets a mutation, then now it's no longer telling your cell not to divide, and so the cell will go on and divide and produce tumors. So that's why they're called tumor suppressor genes. When they're working properly, they prevent cells dividing out of control and producing tumors. But when they become mutated, then the cells go ahead and divide when they're not supposed to. Okay, characteristics of cancer cells. Lack differentiation. That word differentiation just means a cell becoming a particular type of cell. So when after cell division, after a cell divides, that's whatever type of cell it is, whether it's a skin cell or a liver cell or a nerve cell or wherever, it will, when it's first formed, it will not necessarily look like the kind of cell it's supposed to be, but it will soon become a skin cell, and that's called differentiation. Just a cell becoming a particular type of cell. So cancer cells don't do that. They don't become differentiated, so they never function as the type of cell they're supposed to. So if you got cancer cells in your lungs, those cells are just dividing and dividing. They never become normal lung cells. They never become differentiated. They just keep dividing and forming tumors and disrupting the normal lung cells. We have normal large nuclei that's visible under a microscope. So if you're an expert, a pathologist, you can look at a cell and tell if it's cancerous. I used to live across the road from a guy years ago. This was 20 years ago, I guess. And he was a pathologist. He worked at a hospital in a lab. So whenever there was a surgery and something was taken out of a patient, he looked at the tissue under a microscope to see if it's cancerous or not. And even he'd look at blood samples all the time to see if the blood was normal. So there are blood cancers, like what am I trying to think of? What's the more common blood cancer? Yeah, thanks. Leukemia. Yeah, that's a cancer of the blood, and but he had a regular job at a hospital, but he also had a little side business where he had a little lab set up in his basement, just a microscope, and so he worked for some labs that would just give him a bunch of slides every week to take home and look at them, examine them, and to see if they were normal or if they had cancer cells in them or anything abnormal. And he was so good at that. He had looked at millions of slides in his career and said he could look. He said I can look at a cell for 3 seconds and tell if it's cancerous or normal. And so he would get the big box of slides every week, and just in the evening, just for a couple hours, a couple of nights a week, he would just go through these slides, just put one on the microscope, look at it. Oh, that's normal. Next one, that's normal. That's normal. And he'd get paid $8 a slide for that. That was 20 years ago, $8 a slide, and it would take him 5 seconds each to do that. And so he made several $1,000s a week just in a couple hours, a couple nights a week at home doing that. So, if you want to make a lot of money, become a pathologist, but it takes a lot of. You have to have a doctorate degree to do that. So a lot of school. Okay, cancer cells form tumors. Cancerous tissue, so cancerous tissue meaning tumors, they can involve angiogenesis. And this word angiogenesis, it just means growing new blood vessels. In any tissue, growing new blood vessels is called angiogenesis. But tumors, so tumors have their own blood vessels in them to keep them alive, because cancer cells, just like any other cells, they need nutrients and oxygen. So your tumors actually grow their own blood vessels through them. Metastasis, you know that word. When cancer metastasizes, that means it's spread from one organ or tissue to another. Some kinds of cancer are more likely to metastasize than others. Like there's 3 different kinds of skin cancer. The melanoma kind that usually starts out as a mole that becomes cancerous, or often does. That is the least common of the skin cancers, but it's the most likely to metastasize. It very, very commonly metastasizes and metastasizes very rapidly. So melanoma can kill you. It can spread from the skin to other tissues very quickly. So this is cervical cancer, cancer of the cervix, which is the lower part of the uterus. And so these are normal. These are precancerous, so they're on their way to becoming cancerous, and these are cancerous. So the nucleus is large and abnormal compared to these normal nuclei. I guess I couldn't tell. I'm not an expert, but pathologists can look at that and tell. This kind of cancer, cervical cancer, is caused by human papillomavirus. That's the same virus that causes genital warts, so it's sexually transmitted. And then there's a vaccine for that now. Okay, how to not get cancer. So protective behaviors. Don't smoke, of course. Use sunscreen. The sunscreen filters out or blocks the ultraviolet radiation from the sun. Avoid other kinds of radiation like cancer, or X-rays. And be tested for cancer regularly. So that's called screening. Be screened for cancer because, of course, the earlier it's detected, the more likely it can be successfully treated and cured. Or often they say they put the cancer into remission, which means it's no longer active, but they can't guarantee that it's completely gone. And like I was saying, be aware of occupational hazards, chemicals that you might be breathing or coming into contact with, getting it on your skin, or ingesting somehow. And carefully consider hormonal therapy. That's referring to what's called HRT, hormone replacement therapy, HRT. This is what is done for postmenopausal women who've been through menopause, because menopause is the end of the reproductive life of a woman, because the ovaries stop functioning. Essentially, the ovaries stop ovulating, so stop producing eggs that can be fertilized, and also stop producing the female sex hormone. What is the main female sex hormone? Do you know? Estrogen. Estrogen is produced by the ovaries. And so with menopause, the ovaries almost completely stop producing estrogen. And that causes a lot of the unpleasant symptoms or changes in a woman after menopause. Some women have hot flashes and other symptoms. Also, the estrogen, along with works with other hormones to stimulate a lot of normal body functions. For example, your bones. Your bone tissue is always constantly being rebuilt, broken down and rebuilt throughout your life. It's called bone remodeling. It's normal, but it requires estrogen. So after menopause, you have less estrogen, so you're more likely to get osteoporosis because your bones are not your bone tissue is not being rebuilt anymore like it normally does. So in some cases, a doctor will recommend this hormone replacement therapy, which is just I don't know. But they essentially give the patient, the woman, hormone pills, estrogen. Is it pills or injections? Anybody know? Anybody who's had that? I forget. But so it can help with the symptoms of postmenopause, but it can also increase the likelihood of getting some types of cancer. I think cervical or ovarian cancer, or more likely if a person is on hormone replacement therapy. So you have to decide: how bad are the symptoms that it's worth doing this, even though it might make cancer more likely? The protective diet, vitamins A, vitamin C, limit consumption of salt-cured, smoked, or nitrite-cured foods. That's, I think, referring to stuff like bacon and some sausages, or I don't know, salami, some hams. Smoked, I guess some people smoke fish like salmon, and I guess any kind of meat can be smoked, right? I don't know. I haven't eaten meat in the last 40 years. I don't pay much attention. Vegetables from the cabbage family in the diet. So cabbage, lettuce, that also includes stuff like Brussels sprouts, cauliflower. Moderate consumption of alcohol. So there's been a lot of research findings just in the last few years about alcohol. Alcohol is much worse for us than it was thought. I mean, even 10 years ago, it's always been known that alcohol can, can damage your liver. It kills off brain cells. Um, but it's been found that there are more and more problems caused by alcohol, including cancer. It makes a lot of cancers way more likely, so it's a risk factor for cancer. And maintain a healthy weight. Just eliminating all meat or animal products. A vegetarian diet drastically reduces the risk of all colon cancers. Effects of nicotine. Tobacco. Nicotine is a toxin. It's actually a poison. And I mean, if you would just chew a bunch of tobacco and swallow it, it would kill you. You could die from that. I remember when I was a kid, I don't know, 11, 12 years old or something, in the back of a magazine it said, mail in this coupon and we'll send you a free chewing tobacco. It was mail pouch chewing tobacco. And I thought, why not? It'd be free. So I did that, sent it, and a few weeks later, it was a whole big pack of chewing tobacco. I knew nothing about chewing tobacco, never known anybody who chewed tobacco. So I took a mouthful of it and started chewing it, and I didn't really like it, but I didn't know you're supposed to spit it out. And I actually swallowed that stuff. I never puked so hard in my life. I was so sick. My head was spinning. I just about collapsed. It was horrible. Maybe that's why I've never smoked. I never had any interest in tobacco after that. That was disgusting. But anyway, so nicotine is a natural toxin that's that some plants like nicotine, like tobacco plants, they produce that nicotine because it's toxic to herbivores, mostly insects that would try to eat those tobacco leaves. They take a grasshopper takes a couple of bites of that tobacco leaf, and it'll fall over dead, or it'll make it very sick. But anyways, effects of nicotine increase or impedes apoptosis. So if you have some cells that are becoming cancerous, they will be less likely to kill themselves to prevent the cancer developing. And that's a bad thing. So increases angiogenesis, which is okay in normal healthy tissue to have more blood vessels, but if you get cancer, this will help your tumors stay alive, because it'll promote blood vessels growing into the tumor. Activates growth factors, which is okay, except when you got cancer, and now you've got growth factors causing your cancer cells to grow faster. That's not good. Promotes tumor growth, probably because of the growth factors. And impeding apoptosis. And laboratory lab experiments with mice have shown that having nicotine in the body causes tumors to grow twice as fast, twice as large, 9 times metastasis. So cancer metastasizes 9 times. That's 9 times the rate with nicotine as without it. And if tumors have been removed surgically, they're three times more likely to recur or grow back if there's nicotine. So a lot of reasons not to smoke or use tobacco. I've known so many people, so many guys who just cut their lives short by smoking, and so many guys who really loved life. And the guy I worked with, Stuart Valentine, his desk was right next to mine when had a job 20 years ago or so, and hear him on the phone every day. He'd call his wife or his daughter on the phone every day, and you could tell he really loved his wife and kids and talked about them all the time. But he smoked. He smoked. He'd always be going out for a smoke break, and he'd cough all the time. He had this real wet cough, and you could tell he wasn't very healthy. But he got cancer, lung cancer, and it ended up killing him. Lived a pretty miserable life for another year of dealing with lung cancer, but he died. He was in his 50s, I think. Didn't get to even see his daughter get married or anything. There's one guy, another job when I was in my 20s. Real nice guy I worked with. He was in his 60s, and he always had a cup of coffee and a cigarette all the time. And was in a staff meeting once, and somebody mentioned somebody else in the company who retired a year ago just died. Just retired a year ago, and now he's dead. And somebody said, that happens so often. A guy that retires, and then just a year or two later, we hear that he died. And this guy, Doug, who always had a cigarette, he had with a cigarette in his hand, he said, "That's not going to happen to me. I'm going to retire, and I'm going to have a nice, long life." He died before he even got to retire. He had a heart attack. He was in his early 60s, probably from smoking. Never even got to retire. Okay. Okay, so we've been talking about mitosis or mitotic cell division. So we've been talking about eukaryotic cells, like the cells of your body. They divide by mitotic cell division. But what about prokaryotic cells, like bacteria? They divide the same kind of way to reproduce. They divide in half to make more cells, but we don't call it mitotic cell division or mitosis in prokaryotic cells. We call it binary fission. Binary means to, fission means to split in half. And we don't call it mitosis because mitosis involves those 4 steps: prophase, metaphase, anaphase, and telophase. Like in prophase, the nuclear envelope disappears. But in a prokaryotic cell, there is no nuclear envelope. So they can't be prophase. In metaphase, all the chromosomes line up in the middle. There's only one chromosome, so how can one chromosome line up? So we don't call it mitosis, we just call it binary fission. It's much simpler. The one chromosome is copied, and then the cell divides in half.